It is a great pleasure to introduce our next keynote speaker, Professor Manolis Kellis. And I will talk briefly about your biographical background before we can get started with the keynote, just as an introduction. So, Professor Manolis Kellis is a professor of computer science and artificial intelligence at MIT and leads the MIT Computational Biology Group at MIT CSAIL and the Broad Institute of MIT and Harvard. His research seeks to understand the mechanistic basis of human disease, to develop new therapeutics that reverse disease circuitry, and to enable personalized medicine using AI and machine learning to integrate genetics and genomics, and also single-cell epigenomics and transcriptomics, and high-throughput experiments applied to Alzheimer's, obesity, cancer, schizophrenia, cardiovascular, and immune disorders. Manolis has helped lead several large-scale genomics projects, and those include Roadmap Epigenomics, ENCODE, GTEx, and comparative genomics projects. He has also authored over 280 journal publications, cited more than 160,000 times. He received many awards, including the US Presidential Award for Science and Engineering by Barack Obama, the Mendel Medal for Outstanding Achievements in Science, the NIH Director's Transformative Research Award, the Argo Science Award by the Hellenic President, the Boston Patent Law Association Award, and many others. He also received the Sprouse Award for the best PhD thesis in computer science at MIT when he did his PhD there. Manolis has obtained more than 20 multi-year grants from the NIH, and his trainees also hold faculty positions at Stanford, Harvard, and other top universities. He has lived in Greece and France before moving to the US, and he studied and conducted research at MIT, at the Xerox Palo Alto Research Center, and the Cold Spring Harbor Laboratory. Today, he will speak to us about the AI frontier, genomic medicine, healthcare, human knowledge. And before I hand the virtual microphone over to you, I'd just like to remind everyone to put any questions that you may have into the chat or Q&A, and I will pass them on at the end of the talk. And with that, Manolis, thank you so much for being here. Really excited for your talk, and the floor is all yours.
Thank you so much. As I mentioned in the chat, I really enjoy seeing actual humans. I'm sorry for not being there in person, but if you are comfortable, please turn on your video, because by seeing your reactions, I can kind of see who's following, how you're thinking, what you're puzzled about, and so on and so forth. So truly appreciate for those who've already done it. And again, I wanted to say a huge thank you to you, Tim, and to the organizers for having me here today. It's just a true honor to speak to you across the Atlantic. And to see so many young people just taking over science and the next frontiers of medicine is, frankly, I can't think of a better time to be a student right now. I can't think of a better time to be in science and to be, really just transforming the world with everything that's happening. So what I'd like to tell you about today is the work that we're doing in using AI to understand biology, to understand medicine, and to go from systems views of human genomics and human genetics to ultimately therapeutic development and really designing the next generation treatments, the next generation drugs, the next generation approaches to medicine. So I'm a professor at MIT in the Computer Science and AI Lab, and I'm also a member of the Broad Institute. So if you look at medicine, and this is, a school for medicine, for AI and medicine. So medicine has really come such a long way. Basically, we went from looking at patients from the outside to being able to see inside the body and eventually inside the cell. And more than 100 years ago, Alois Alzheimer diagnosed what was previously known as simply dementia, of elderly people losing their mind, quote unquote, to a disease that is very clearly pathological, that has a very clear cellular signature. And that cellular signature that was first discovered more than 100 years ago is still the way that we diagnose Alzheimer's today, namely the two signatures of amyloid beta plaques in the intercellular space and neurofibrillary tangles within the cells. But we have come a long way even since then, from the body to the cell to the subcellular, to now start looking at individual genetic letters that are associated with disease and that are in fact playing causal roles in disease. If you look at APOE4, for example, the strongest genetic association with Alzheimer's, the E4 allele of apolipoprotein E gives you a 10-fold higher risk for Alzheimer's compared to controls, which is quite dramatic. And the the key concept of this correlation with genetics is that there's a causal underpinning. Namely, because we inherit these genetic variants at birth, it's not because of my lifestyle or because of my diet that I inherit the disease variants. This is actually playing a causal role. So basically, we're going from an observation to suddenly causality and this extraordinary resolution of individual nucleotides. However, the challenge of genetics is that, yes, it's all powerful and it can tell you that something is indeed playing a causal role, and it is mechanism agnostic. In other words, you can discover a genetic association even without knowing how it acts. But that blessing of genetics, that it works even without a mechanism, is also the curse of genetics in that after you discover the association, you have really no idea how it works. You sometimes don't even know what the causal gene is, what the right cell type to look at is. And that's where all of the functional genomics comes in. So on the left side here, I'm showing you 4 cells and 2 phenotypes of plaques and tangles. On the right side, I'm showing you 2.4 million cells across 430 donors, where every dot is a different cell in these 2.4 million cells, and the position of that cell relative to all of the other cells is dependent on 20,000 variables, not 2 variables, namely the expression of each of the 20,000 genes in that one cell. So this is an extraordinary time to be in medicine, to be in biology, to be, frankly, in AI and science more broadly, where we have undergone all of these different transformations building on each other, and we are now here with extraordinary tools at our disposal that we can now start integrating. And that integration is very much relying on AI. This combination of data, of genomics, is very much enabled by AI. And there are 3 transitions that I want to walk you through that have enabled this new era. The first transition is we're going from a hypothesis-driven research to data-driven research. Classically, you would formulate a hypothesis, go and gather a lot of data. You would do a lot of thinking before, and then design a targeted study that would give you a yes/no answer to your hypothesis. You know, yes, it is still supported, or no, you can reject that hypothesis and work on a better hypothesis. But the problem is that this is a highly biased approach, and allows for very little novelty. By contrast, we can now switch to this data-driven research where we can basically shoot first and ask questions later. We ask it, we gather a ton of data, minutely measuring thousands of variables in the system, sometimes millions of variables. And then we ask the questions once we have the data. So basically, the idea is that we want to build systematic data sets, build these massive resources, enable massive data sharing, have comprehensive use of biology, and then outcome, not just 1 or 2 answers, but thousands of new hypotheses, models, and ways of probing the system that were previously unfathomable, and outcomes a lot more novelty, because now you can go in places that you wouldn't have even suspected before. The second transformation is going from correlation to causation. Correlation has been the basis of epidemiology for the vast majority of human condition. You would observe that some things correlate with others. And, nowadays, for example, you can say, oh, great, more coffee consumption correlates with better health. If you, if you drink a $7 Starbucks coffee every morning, you also have better health outcomes. Is it because Starbucks coffee is so great? Or is it because the folks who can afford a $7 coffee every morning are also the ones who can afford, I don't know, membership to a gym, better healthcare, better nutrition, better lifestyle, etc. Similarly, countries that consume more chocolate also have more Nobel Prizes. Is it that chocolate and coffee are really just extraordinary? Frankly, it might be. But, there's another possibility, which is that it's just correlation, not causation. But with genetics, you have now the ability to start predicting disease outcome, to start computing polygenic risk scores, start inferring causal factors. And you can also, in biology, have perturbation experiments in cellular systems and in animal models that allow you to confirm these causal networks. Whereas in social studies, for example, doing an experiment where you, ask a whole population to do something different can be very difficult and very costly. And of course, human experimentation is just not ethical. So you can't carry out many of these things that you would like to in other fields. But in biology, you can intervene both in cellular systems and in animal models. And then the third, and perhaps the most major transformation of all, is what has happened in AI. Basically, we've gone from this classical machine learning and classical data analysis, where you basically develop a new methodology for every problem, where the human scientist would do all of the thinking, and you would sort of build a new program for every new data set. And you would try to have as few parameters as possible and to have very targeted models, to the transformation that's happening right now with generative AI and deep learning, where we are now able to build foundation models, that we're able to build models that understand multiple modalities, understand images and text and knowledge graphs and protein structure, and that truly understand these concepts. So very often I hear people say, oh, yeah, this is something that we've had since the 60s. Neural networks have been around. The only difference is that we now have bigger models. And I don't agree at all. There's, yes, bigger models. Yes, there's more data, but there's a fundamental shift that has happened, which is the concept of representation learning. The concept that I can now give billions of parameters rather than try to limit the number of parameters. I can give billions of parameters to my model. And my model can basically learn from itself. It can basically do self-supervised learning. For example, you can take thousands of images and, thousands of millions of images. And instead of having humans curate every single one of these images, you have the machine hide a small part of the image and then try to repredict it. So the machine is giving itself small challenges. And in trying to repredict the image, it can get, somewhat good by understanding that there's gradients and continuity in the colors. It can get a little better by understanding the roughness of the background and so on and so forth. But to get amazingly good, to get like 60%, 70%, 80%, 90%, 100% accuracy in reconstructing the missing parts, you truly need to understand what the world actually looks like, to understand the continuity of the world, to understand the discreteness of objects, to understand which objects go well with each other, and so on and so forth. And this is the same thing, not just for images, but also for text. When you're looking at ChatGPT, for example, a lot of people are saying, oh, great, ChatGPT was basically trained on next word prediction. What do I care about the next word? And the answer is no, that's a pretext task. This task is there to train the model to understand human thought, to understand human language, to understand human emotion, to understand the human psyche, if you wish. Because to get really, really good at next word prediction, you need to understand the topic, the theme, the language, the style, the everything about human thought. And that representation learning, that self-supervised scenario, these multimodal, deep, hierarchical, nested layers of abstraction then leads to these foundation models that can have emergent capabilities. They can truly understand these concepts. So what I'm going to talk about in this talk is basically, number one, how do we carry out these, first two components of going from hypothesis to data-driven, from correlation to causation. And I'll give you a few examples about single-cell genomics and how it helps us understand cellular circuitry. And then I'm actually going to switch to the second part here to show you the extraordinary things that we can do with representation learning and generative AI, where we can basically start understanding the language of protein folding together with English language in a multimodal model, and use that to start transforming the way that we design therapeutics, the way that we understand targeting of complex traits, and so on and so forth. Okay? Can I get a thumbs up from those who are following? Are you guys with me? Yes? Awesome. Good. Okay, so how do we dissect the mechanism of disease-associated studies? So as I mentioned earlier, we start with genetics. We start with genetics because through these both common and rare variants that are associated, correlated with disease, we actually have causation because of the spatial nature of genetics, which is not a consequence of the environment, but instead causing the disease. So that causal nature basically allows us to say, if we go into these regions and we understand the mechanism through which the genetic variants are acting, that gives us points of intervention. So that's the first step. The second step is to then complement the genetic data with molecular and functional genomics. What does that mean? That means that instead of just saying, oh, there's a variant here, I'm going to look at hundreds of tissues in the human body, thousands of cells from each person, hundreds of cell types across people. And I'm not just going to look at RNA expression, I'm also going to look at the epigenome, specifically DNA accessibility, histone modifications, DNA methylation, that modulate the interpretation of the primary sequence of the human genome. And I'm going to do that both in healthy and in disease samples to understand the functional differences at the molecular level, both as a consequence of genetics and as a consequence of the environment, behavior, nutrition, and also disease itself. This gives us the big data that I was talking about earlier. This gives us this, systematic data sets that allows us to have comprehensive views. And that's where the fun begins. You can then start integrating these data sets using convolutional neural networks to understand regulatory motifs, using graph neural networks to understand knowledge graphs and emergent properties, to understand regulatory networks, using statistical genetics, using causality analysis and mediation analysis, and other approaches to really predict what are the driver genes, control regulatory regions, the regulators that bind these regions, and the cell types and pathways that are implicated. Once you have that, you can then turn to validate these predictions, both in human cells and in mouse models. That's where the perturbation experiments come in to confirm what we postulate as causality by combining genetics and functional genomics. And of course, we disseminate the results and start the cycle all over again. Now, what do I mean by these networks? Let me dive right in. So here's a region of genetic association with cholesterol. And what you can see is that there are exactly 4 genetic variants in this region that are correlated with the disease and therefore likely to play causal roles, at least one of them. What we can now ask is, from these genetic variants, what are the control regions where these variants are likely to act? How? By looking at the epigenomic information, by looking at the histone modifications and the annotation of the language of the epigenome. And we have developed a series of methods through the Roadmap Epigenomics project, through the ENCODE project, through the EpiMap project, to systematically understand these histone modification combinations. We've developed programs such as Chrome-HMM with Jason Ernst to infer a small number of chromatin states from the combination of these histone marks. That allows us to now start annotating enhancer regions, these long-range control regions that are acting both upstream and downstream of different genes. And to use that to both distinguish, of course, enhancers and promoters and transcribed regions, but to also find what are the cell types and states where these enhancers are active or repressed or poised, and so on and so forth. So we can use this functional information to go from just a region of association where we have no idea where it might act, to understanding what are the enhancers, what are the cell types, what are the upstream regulators, what are the regulatory motifs that are sometimes disrupted either directly or indirectly by the genetic variants, and ultimately to understand what are the target genes that are altered by this genetic lesion, what are the cell types and pathways where these genes are acting, and ultimately make a prediction for the mechanism underlying the disease. Let me give you one example. In collaboration with Melina Klausnitzer and this extraordinary team of collaborators, we basically studied back in 2015 in the New England Journal of Medicine as part of the Roadmap Epigenomics project. We studied the strongest genetic association with obesity in the FTO locus. And we basically said, okay, this region is causing 1 standard deviation higher weight in the carriers who are homozygous for the risk allele, similar to how APOE4 causes a 10-fold increased risk. So these are somewhat of an exception because it's very rare for a genetic variant to be both strong effect and common. This one is actually carried by 42% of European chromosomes, 44% of Southeast Asian chromosomes, but only 2% of African chromosomes. So the question now is, how is the variant acting? And what we found by combining all of these different approaches together in this paper is that you can trace a 50,000 nucleotide region of association down to a single letter. A change from a T in this AT-rich motif recognized and bound by the ARID5B repressor, which is the AT-rich interaction domain protein ARID5B. So that repressor normally binds this motif, but when the T changes to a C in the obese risk individuals, what you see is that the repressor can no longer bind, and you have this overactivation of this massive 12,000 nucleotide long enhancer region that is it turns out is a master regulator of IRX3 and IRX5. These genes are not just sitting right next door. These genes are actually 1.2 million nucleotides away and 600,000 nucleotides away from the causal variant, several genes over. So that basically tells us that we can't simply just look under the lamppost to say, hey, what's happening in the FTO gene? Notice that the FTO gene doesn't even feature in this circuitry. It is just a red herring. Its name was initially fused TOS-O, and it doesn't have any obesity phenotypes if you're very cautious not to perturb this region, this control region, when perturbing the gene. So great, we now have 2 other genes, IRX3 and IRX5. What do? What they do is that they are master controllers of a process known as thermogenesis, which is the generation of heat. It's a form of browning of mitochondria, where basically you depolarize the mitochondrial membrane. You cause a loss in the proton gradient that normally is utilized to contract ATP and to store energy into lipids and eventually into white adipocytes. And thermogenesis, controlled by uncoupling protein 1 and PGC1 alpha and PRDM16, which are all downstream of IRX3 and IRX5, what these genes do is that they decide to, instead of storing the excess calories in your diet, the ones that you don't burn with exercise, you can decide to store them in white adipocytes, or you can decide to burn them in brown adipocytes. And the reason for this is this developmental transition. Early in the first 3 days of differentiation of these adipocytes, the carriers of the C allele are unable to turn on thermogenesis. And the carriers of the T allele are able to just simply burn the calories. So basically, the carriers, the risk allele carriers, are in fact storing all of the calories instead of burning them, and vice versa. So why are we so excited about this? I mean, of course, academically, it's extraordinarily amazing to see this region that was mysterious traced down to a very clear mechanism. And that's a beautiful academic exercise. But much more importantly, from a medicine standpoint, we can now start intervening. We have entry points for intervention. We can basically go and increase or down regulate the expression of the upstream regulator or of the downstream target genes. You can also use CRISPR-Cas9 genome editing to establish the causality of this region and edit the C of risk individuals back to a T to restore the motif, or the T to a C to disrupt the motif. And then what you find in every one of these cases is that they work like a switch between lean and obese. You can go back and forth. You can basically take primary adipocytes from risk individuals, the carriers of the C allele that don't have a functional motif. And by changing a single letter from 3.2 billion letters in the genome, you can restore thermogenesis, measured here as oxygen consumption rate, uncoupled as a percent of basal, both in control and in isoproterenol stimulated conditions. And you can see that you can restore the cellular phenotype by a factor of 7 with a single letter change. You can also go into mice and take these genes that are de-repressed in obesity and shut them off. You can basically use an adipose promoter and a dominant negative form of IRX3 to shut down IRX3 in mice. And what you can see is that these mice are leaner to start with, but when you introduce a high-fat diet, what you see is that regular mice gain weight, but these mice are simply unable to gain weight. They don't change their exercise, they don't change their diet. What they do is that they're simply burning more calories when they're awake and when they're sleeping. So this is one such example, going from a region of association to a letter, a regulator, downstream target genes, a mechanism, a cell type, and ultimately the ability to intervene. That's what we want to do systematically. That's what we want to do for every one of the 100,000 genetic regions that are robustly associated with human traits and phenotypes and with human disease. So I'm going to walk you through a couple more examples. So this is the strongest association with Alzheimer's that I mentioned earlier. So this is the APOE gene and the E4 allele versus the E3 and the E2 allele. And what we did is that we basically looked at carriers of the E4 allele, whether they had developed Alzheimer's symptoms or not, both male and female, and with and without pathology. And what we found is that the carriers of the E4 allele showed decreased myelination. So if you look at the axons of neurons, what you see is that in control individuals, in E3 and E2 individuals, they have this nice thick myelination. But in E3 individuals, you have much less myelination. Now, what's funny is that myelination, of course, depends on lipids. This is like lipid-based, and specifically cholesterol is transformed into myelin by the oligodendrocytes that then deposit the myelin surrounding the axons. But if you look at cholesterol biosynthesis pathways, you see that they're actually upregulated in oligodendrocytes. So this is really weird. So why would you make more cholesterol, but in the end up with less cholesterol? And the answer is exactly here in the middle. Basically, the E3 individuals, when you look at their cells and their oligodendrocytes, you see this nice endoplasmic reticulum striation, which is a sign of a healthy cell exporting whatever it needs to export. By contrast, in the E4 carriers, even for heterozygous carriers, what you see is these bubbles instead, where this accumulation of these lipids, of this cholesterol, is found in the oligodendrocytes. So our hypothesis was that perhaps this is getting upregulated because of transport problems. You cannot transport it in the right place. And indeed, by using cyclodextrin, this hydrophobic molecule, to restore and facilitate cholesterol transport, we saw that we can restore myelination both in human cells and in mouse cells. But even more importantly, you can restore cognition. You can help mice recover their lost memories. So this is another one of those examples where we can basically go from a region of association to a specific mechanism and ultimately a therapeutic intervention, which again will take many years to translate to humans, but is very promising both in human cells and in mice. In the third example, I want to take you through cancer immunotherapy, specifically anti-PD-1 immunotherapy for metastatic melanoma. So metastatic melanoma used to be a death sentence, and the individuals with it would have very, very few months, and if they're lucky, years to live. But with immunotherapy, 50% of the patients are basically completely cured because you can restore the immune system, and the immune system can now start attacking the cancer. But what makes the other 50% of individuals not respond? We basically looked at responders and non-responders, and specifically we looked at H3K27 acetylation, a mark of enhancers, in the context of multiple histone states or chromatin states. And what we found is that for a large number of genes, the responders and the non-responders were differentially active in those enhancer regions. We used that to start predicting the regulatory motifs, the binding of regulators in these regions, and we predicted a regulator that we could use as a combination treatment along with anti-PD-1 therapy to restore the epigenomic state of responders and non-responders. And what you can basically see is that with that combination treatment, the inhibition of this acetylation reader bromodomain combined with anti-PD-1 reduced the tumor volume, where instead of seeing 50% response, we saw 100% response. And then this is in collaboration with Jackie Yang, David Liu, Kunal Rai, and Geneviève Bolland, an extraordinary team of people. So this is a third example, and I want to sort of emphasize this extraordinary transformation that we can now go from hypothesis and correlation to data-driven and causation. And I want to give you a few examples of the insights that you can gain from this data-centric approach. So this is an example from a paper that we just published a few weeks ago, looking at 430 donors, 6 brain regions, 2.4 million cells across prefrontal cortex, mid-temporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body. In the initial study back in 2019, we had looked at 24 control individuals that didn't show any pathology and had high cognition, and 24 both early and late stage AD individuals. And what we found is that the early changes in Alzheimer's were extremely cell type specific, but the late changes were much more cell type shared. We also found that there were massive differences between male and female individuals. The female individuals showed cells that were much more Alzheimer's-like than the male individuals. And we also found that the differences between male and female individuals were large to start with prior to Alzheimer's, but even larger with Alzheimer's. So we have 3,000 differentially expressed genes in excitatory neurons between men and women, but 6,000 in the context of AD. And in particular, we found that female individuals showed less upregulation of myelination pathways that I mentioned earlier, and showed, in fact, more white matter loss in their brain pathology, suggesting that perhaps we should be thinking about sex-specific interventions for many of those. Again, this early study was across 24 cases and controls. We have now expanded to 430 individuals. This is a paper that was published in Cell a few months ago. And what we can now do is start asking about the diversity of phenotypic variation. We can look at diversity in pathology between, again, amyloid, plaques, and neurofibrillary tangles. We can also look at the diversity in cognition between episodic memory, semantic memory, perceptual memory, working memory, and so on and so forth. And of course, global cognitive function. And we can also look at the decline, not just where's your cognition today, but how fast has it been going based on your age. And then we can use all of this to start defining both signatures of resilience for the neurons, namely what neurons are preferentially lost and which neurons are preferentially kept. And you can see here several inhibitory neuron subtypes that are specifically selectively vulnerable. And you can also use that to start asking about what are the major hallmarks of Alzheimer's disease across mitochondrial dysregulation, mRNA metabolism, cohesin, lipid metabolism, etc. And what we find, for example, is that, as I mentioned earlier with cholesterol, we find all of these lipid metabolism pathways are dramatically altered in the case of Alzheimer's. We can also use this to look for individuals that are resilient, namely individuals that show strong pathology, but very little cognitive decline. And those individuals are basically showing this dramatic change in this choline metabolism pathway, which appears to be protective. To give you more context, what we found is that in astrocytes in particular, which are these helper cells that help recycle neuroreceptors, that also help nourish the neurons and take care of them, we basically studied the modules that you can uncover by looking at gene-gene correlations and projecting the gene correlation matrix into a smaller number of modules, and then understanding what are these modules in astrocytes. We found that there's a small number of modules, very specifically in astrocytes, associated with cognitive resilience. And then this pointed us to choline metabolism as one of the major protective pathways. We can also go a step further and start asking for subtypes of microglia. And in 2015, we basically showed that the strongest genetic associations with microglia, with Alzheimer's, were localized very specifically in microglial enhancers and also macrophage enhancers, implicating immune pathways in Alzheimer's. And what we can see now is that the state of microglia changes from these inflammatory microglia in the Alzheimer's individuals to the homeostatic microglia in the control individuals. And the question is, can we prevent this transition to inflammatory by turning on the homeostatic regulators? And what's really interesting is that it takes a very small number of regulators to go to inflammatory, but a large number of regulators to go back to homeostatic. And by overexpressing some of these regulators, we can basically see that indeed we can restore this homeostatic state, suggesting again a treatment that focuses on the cellular state rather than individual genes. Another type of analysis is to look at very specifically what are not just the gene expression changes, but what are the mutational differences between cases and controls. And in particular, we all start from a single cell, which basically undergoes a series of divisions that ultimately give rise to all of the cells in our body. And what we found is that you can now use the single cell RNA data to start inferring somatic mutations that might be happening in those lineages that might be affecting one or more cells. But because we're looking at RNA, we're capturing both somatic DNA mutations, but also RNA mutations that might be due to all kinds of environmental and other toxin environments, for example, oxidative stress. And what we found is that the Alzheimer's individuals show a much higher burden of this pathology than the non-Alzheimer's individuals, and that this pathology localized very specifically in pathways associated with energy metabolism and a few others. We also found this global epigenomic alteration that is happening between cases and controls, where the entire epigenome appears to be flattening out, which is a process that we call epigenome erosion in these late Alzheimer's individuals. We're also finding a lot of insights in other neurodegenerative conditions. For example, if you look at ALS, amyotrophic lateral sclerosis, associated with motor disorders, you find that it is dramatically different from in its phenotype with frontotemporal dementia that instead manifests as a cognitive and personality disorder. But even though they're so different phenotypically, they actually run in the same families. And very often, the C9orf72 carriers of the same family will sometimes manifest as one disorder or another disorder in the spectrum of both TDP43 and tau pathology. And when we looked at the single cell level, in the motor cortex, which is affected in ALS, and in the frontotemporal cortex, which is affected in the frontotemporal dementia, what we found is that the changes were nearly identical and the cell types were nearly identical. Namely, if you look at the prefrontal cortex and the motor cortex, there's nearly a one-to-one correspondence between the cell types, and the genes that are altered were very specifically these long-range projecting neurons that we call von Economou neurons in the frontal cortex and BET cells in the motor cortex. So these suggest that, in fact, there might be a common mechanistic basis between these two seemingly very different disorders. We can also gain insights in schizophrenia and psychiatric disorders. So in a paper that was just published in Science a few weeks ago, we can basically see that the differentially expressed genes help us understand a large number of the genetic loci associated with schizophrenia. And very importantly, we can actually start using the transcriptional signatures of these individuals to distinguish those donors where their post-mortem brain expression looks very schizophrenia-like, and those where the post-mortem expression looks very Crow-like. And this was very consistent with the phenotypic annotations. You can see here a lot of schizophrenia cases were on the right, and a lot of controls were on the left. But there's some striking exceptions. In this control 7 individual, you basically see that their transcriptional profiles are nearly perfectly schizophrenia-like. But then that individual was a control individual. Control with a star, however, because the son of this father was in fact diagnosed with schizophrenia. And, this has a prevalence of 1 in 1,000. So you can infer that maybe there's something going on pathologically or cellularly or molecularly for this person, and that they were perhaps not diagnosed. And, in the next generation, you can kind of see this manifest. And conversely, those individuals who were schizophrenia cases, but control-like in their transcriptome, showed an excess of this SCTR state in their transcriptome, suggesting perhaps there's another pathway to schizophrenia for those individuals. We can also gain a lot of insights in addiction by looking at what are the changes that are happening very specifically in specific subtypes of the nucleus accumbens that are associated with different dopamine receptors. And there's a lot of synaptic, mitochondrial, and ribosomal changes, and a lot of AP1 family regulators are in fact coming up. And you can also start asking about differences that are affecting multiple tissues simultaneously. For example, we know how extraordinary the effects of diet and exercise are across the entire body. So we looked both in humans and in mice at this diversity of muscle, fat, brain, liver, heart, and blood. And what we found is coordinated changes across many of these cell types, very often mediated by immune cells and these adipocyte progenitor cells. And in particular, I want to tell you about a new way of thinking about phenotypic variation in these individuals. Traditionally, we have a single annotation that this person has Alzheimer's disease, and this person doesn't have Alzheimer's disease. But with single cell data, you can go a step further. You can basically take the neighborhoods of cellular states across the 430 individuals and start painting every cellular neighborhood, which are discovered based on simply the transcriptional state of those individuals. You can take every transcriptional neighborhood and ask, what are the neighborhoods that are enriched in cells from Alzheimer's individuals? And what are the neighborhoods enriched in cells from non-Alzheimer's individuals? So for every neighborhood, we get an enrichment score as to whether it's Alzheimer's or not. And you can now take that score and ask for every cell, is it more Alzheimer's-like or non-Alzheimer's-like, regardless of the annotation of that individual for that cell. Does that make sense for everyone? So that basically means that I can now ask for a given person if their microglia might be more Alzheimer's-like in their transcriptional locations, and if their oligodendrocytes might be more control-like. So I'm not constrained anymore to the phenotypic annotation of the person. I can now start thinking about the cellular diversity of that person by projecting the phenotypes of this individual onto their cells, and using the cell projected phenotypes, understand the heterogeneity of each person. And you can now see how these neighborhoods are basically going from a healthy neighborhood to, in blue, to disease neighborhood in red. And every cell falls somewhere in that neighborhood. And initially, this can be quite noisy, but the moment you start aggregating across these hundreds of donors, this basically gives you an extraordinary robust score for their microglia pathology, for example, for every cell, which you can then aggregate for every person. And you can see that the Alzheimer's-like score is much more severe for the Alzheimer's diagnosed individuals than for the control individuals, but you also see that there's a spread. Every person has both healthy and disease cells. And you can now start using that to start mapping these phenotypes at the cellular level and to start uncovering the corresponding regulators, cell types, and pathways underlying this. But much more systematically, I want to start thinking of individuals in a more rich way. Instead of just saying Alzheimer's and control, I want to say, yes, sure, Alzheimer's, but what pathways, what hallmarks of Alzheimer's does that person show? In the same way that we have been talking about cancer hallmarks for more than 20 years now, I think it's time that we start thinking about these hallmarks in every disorder. And for Alzheimer's, we can start thinking about, translation dysregulation, synaptic dysregulation, GTPase, oxidative phosphorylation, iron, and start associating each of these hallmarks with distinct phenotypic differences between those individuals. And also start painting individuals into groups and start finding subtypes of Alzheimer's disease associated with distinct combinations of these pathways. And we can look much more systematically within the clinical records of these individuals to start asking about causal mechanisms that might be leading to the progression of different diseases. And in particular, we've started building these large language models for interpreting these changes mechanistically. So we have built this generative additive model for distinguishing this response curve across individuals between, for example, creatinine, a biomarker of disease, with mortality odds, and then distinguishing this by breaking up this curve. And we can then start interpreting each of these components using these foundation models. So this takes us basically to now this generative AI and deep learning revolution. What is actually happening here? How can Gen AI help us cure disease? How can it help understand the mechanism of disease? And the key insight that I want you guys to understand is this difference between the original data, which we can call X, these are the pixels in the image, the Y, which is the annotation of the image, is it a dog, is it a boat, is it a cat or a bird, and this latent representation, Z, which is the machine interpretable representation of the corresponding inferred high-level abstractions that you can extract from that image, okay? So that's the key idea of deep learning, that you're going from pixels to features that are hierarchically built, and these features are inferred by the machine at every iteration. So through these convolutional operations, you can infer filters or patterns, and some patterns correspond to vertical lines, others to horizontal lines. And by combining these patterns, not the original pixels, you can infer what a wheel looks like or what a face looks like, and so on and so forth. And you can build that into a hierarchical representation. And we have been using this in biology to start building hierarchical representations of regulatory motifs in the genome. starting from the nucleotides and then inferring more and more complex abstractions that allow us to start predicting the class of a given region, if it's bound or not bound by a particular regulator. And you can do this not just with images and genomes, but you can do this in graphs representing chemistry. And you can basically have abstractions learned for every atom that initially depend only on that atom, and then on the neighbors, and then on the neighbors of the neighbors, and so on and so forth, to learn more complex representations by going up the hierarchy of increasingly abstract concepts. And you can do the same thing with language, where you can start with individual words based on their context, mapping them with word2vec in these latent embedding spaces of individual words. And you can combine these words together across sentences to start understanding through transformers the meaning of entire sentences. And you can use that to now learn jointly both text and images through these joint embeddings, these multimodal, multidimensional embeddings of both human text and natural images. So we've used this approach in collaboration with Marinka Zitnik at the Harvard Medical School to basically co-embed protein folding and biomedical text description of the function of these proteins in this combined model that we call TXPLM, and more recently Proteus. And using this, we can now start inferring these domains and their meaning, and we can start asking questions to the system. We now have a communicative, interactive program that allows us to now reason about protein structure. And we can plug in the same concept on graph neural networks of knowledge graphs, where every node here is a gene, every node here is a phenotype, every node here is a disease. And you can start connecting these nodes together in PrimeKG graph neural network that was built by Marinka Zitnik. And we can now start asking about the latent representations of every one of those nodes. And you can see how diseases are now starting to be pulled apart into different clusters, and genes and proteins and drugs and pathways. And we can now start correlating the protein structure of these proteins with the function as inferred by this latent embedding projection of the graph neural network interpretation of the knowledge graph. We can basically now start learning multimodal representations from these knowledge graphs and start mapping protein structure to those representations. And I mentioned earlier that we can basically learn these latent embedding projections of individual cells and map phenotypic variation onto those cells, again, learning these multimodal embeddings across millions of human cells of both phenotypic variation and transcriptional variation. And you can do the same thing where the dots are not cells anymore, but now the dots are individuals. And you can look in this particular two-dimensional projection of phenotypic variation across pathology and cognition, that the two are in fact falling in different axes. And that the genes that are differentially expressed along with the cognition axis are expressed in excitatory neurons, but the genes differentially expressed in the pathology axis are instead in microglia and other oligodendrocytes and astrocytes. And you can use this to not only map data, but also map text itself. So for example, collaborating with a group that's looking at the responses to how people are feeling, you can now start creating a latent representation of the responses that people are giving as to sort of how they're feeling. You can basically build an embedding map of emotional wellness. You can do the same thing with simply the Swiss prot descriptions of protein function. You can basically take the description of every gene in the human genome and then create a latent representation of those genes. And you can go and browse these representations. You can basically go to these latent graph embeddings For example, I can go here to search gene, and I can click on these genes. And this is, a program that we've built in my own team. We call it Mantis. And it basically allows us to now go and navigate the latent embeddings of these genes. And every single one of these dots is a different gene. And then you can see how they're grouped by their functions. And you can pull here on the right the corresponding representations, and then you can expand out, particular functions associated with different parts of the space. And what's really cool is that you can now start, searching, for example, for lipids, and then you can see, what are all of the results that come up with lipids. Some are associated with autophagy and regulation, others with phosphatase. And you can also ask questions about these results. And basically, what are the key functions of those genes? And then, the narrator is basically saying, well, let me select first a group of genes, and then you can ask a question, tell me about these genes. So, it's able to think about, sort of what's common between them. So basically, I want you guys to start thinking systematically about what can we do if we have these latent embeddings and we have this ability to navigate this cognitive space. And in particular, you can sort of think about what a student does with a single paper. A student reads a paper, and a student can really truly understand a paper. Any smart student can understand a paper. But the difference is that if you have an expert in that field, that expert, as soon as they see the paper, they know where that paper fits in the fabric of knowledge base. And again, this is a beautiful metaphor of knowledge base as this, sort of Einstein-like dream of what the fabric of knowledge base looks like. It's a great picture of Albert Einstein, but, that's not his quote. I'm just, making this up. But much more importantly, maps allow us to navigate, to strategize, to make differences, to make decisions about how to fight off an army of 10,000 when you only have 300 people with you. And we can use these maps for cognition. We can use these maps for human ideas. We can basically build cognitive cartography. And this is, for example, 3 million articles of the New York Times. And you can create these ontologies on the fly. Nobody had to decide back in 1980 what the ontology of human culture would be. You can decide on the fly. And in the same way, you don't have to decide now what the ontology of mental health will be for teenagers in 2024, when you're in 1980. You're not going to know this until you see the data. So I think this ability with AI to start creating these latent embedding spaces, these Z vectors, and navigate them directly and understand how they play out across different fields. For example, here I'm showing you in education, every class taught at MIT and all of the relationships between them. Here I'm showing you 100,000 loans that are clustered by their description and are showing quantitative information. Here I'm showing you every grant, every paper, and every idea basically generated and published by MIT authors, along with the physical space where those grants and those patents and those ideas are generated in a multimodal way. And here I'm showing you for my own research team how ideas evolving between different students and different topics over time. So the importance of these cognitive maps cannot be overstated. The landscape, when you go skiing, having a map basically means that you have an idea of where you are, where you're going, how the place where you are fits in the larger landscape, and it allows you to abstract, to summarize, to make decisions. And this is what this Mantis platform that we have built allows you to do. It allows you to sort of reveal clusters, allows you to search, to look at the tree, to understand the results in a categorical way. And we've built this narrator and these agents, and we have now an orchestrator that allows you to coordinate these different agents. And we're building this Jupyter Sandbox compute platform that allows the narrator to create this code and to run it directly on the data. And we want these latent embeddings to be directly interpretable, directly mappable, And you can do this across many, many different changes and modalities. So that's where I'm going to stop and go here and basically say that, yes, this can also, of course, be used in education. And we're building now 3D ways of navigating these landscapes and then creating agents in each one of these knowledge buildings, if you wish, to be able to navigate mathematics or chemistry or neuroscience or biology. And that's where I kind of see the path forward. I see the path of science of building these multimodal embeddings to understand protein structure, to understand protein function, to understand knowledge graph, to understand chemical structure, to understand cellular states, to understand patient trajectories, and to start integrating them all in a unified view of all of these different layers of biology. But I also see AI as providing a Google Maps for providing a landscape, a cartography, not just individual points, but how they weave together with each other in this knowledge space, where you can basically have this visual search and interaction and integration, where you can do retrieval augmented generation or RAG in a visual way across millions of documents that you can sort through and then decide what areas of the knowledge you care the most about, with this hierarchical, interactive, direct, visual, transparent, explainable representation and manipulation of that underlying knowledge. So I'm very excited about where the field is going. I'm very excited about sort of the possibilities that are ahead of us. And I'm thrilled to speak with all of you. And I don't know if there's time for questions, but I would be thrilled to hear your reactions. And I can see a few questions in the chat.
Yeah, thank you so much, Manolis. This has been incredibly inspiring and fantastic work. Really. And I think we have time for maybe one question. We're a little past schedule, but I think let's take one or two questions. And maybe start with Krina Samargitin, who had a question on morbid obesity. Would you like to answer
Yes,
that?
thanks a lot for a great talk. And my question is more like it's a fantastic research and results you have. And can you use, or maybe you use it already, this method technology to treat morbid obesity? And then the same, but clinical applications for Alzheimer's disease treatment.
Yeah, it's a fantastic question. So basically, of course, that's our goal. That's exactly where we want to go. Going there is not going to be done in an academic lab. This is exactly the kind of thing that requires collaboration between pharma, between, biotech, taking these technologies, applying them, finding the targets is the first start, but then finding the molecules is the next step. And that's what we're doing now with Brad Penteluta in the chemistry department and Marinka in the Harvard Medical School, to basically start combining these together to not only find the targets, but to understand the biology surrounding them and to understand the chemistry and how to intervene. I see Tilak has his hand up.
Thanks.
Thank you.
Thank you very much. Loads of questions, of course, but one that I was quite interested in, this might be something very obvious to you and some of my colleagues, was the idea of the features that develop in the latent space and, vertical lines, horizontal lines, etc. And that sounds very much like the way the visual system in the brain works, and I guess other networks in the brain. Now, we know we call these neural networks, and they're loosely based on, the brain, I suppose, but I wasn't sure whether that was, on purpose. And is this something that has just emerged as a function of these networks, or was that the design,
Yeah.
the thought behind this in the first place?
What's really spooky is that we think we've hit onto something very fundamental here. When we showed the graph that we have built of PBS episodes, for example, we basically looked at, every episode that had been built by Nova and the documentaries. And we showed this map, and the folks who developed this episode started looking at them, and they said, oh, that's spooky. That's how I think of them. And a few weeks later, we basically see this paper where if you look at how participants are thinking of particular words and what is the proximity in their brain reactions to these words, to the proximity of how the LLMs are thinking about the same words, you found a nearly one-to-one correspondence. And it might not be a coincidence that the way that humans are learning from context is through this constant prediction of what might happen next, and then this adjustment of this prediction based on what we see. And if I tell you the words liberty and freedom, and to use them in a sentence, you would use them in a very different way in different sentences. But I would be hard-pressed to define what makes these two words different. They mean the same thing, but we use them differently. And maybe we learn about the context of these words the same way that LLMs learn about the context of these words. So we may have hit upon something very fundamental about the way humans encode knowledge through this association machine and this representation learning that we have. I see Jacob is next, or Jacobus, rather. And Simon, can you guys turn on your video so I can see as well? Thank you.
Sorry to interrupt. Just to be mindful, I think we have to be really quick with these questions.
Okay, sounds good. I'll be super quick in my answers.
Okay. A quick question. It was fascinating when you showed the distribution of the single letter change in adipose cells. And that suggests that it was probably somewhere on the migration out of, sorry for the background noise, the migration out of Africa, that there may have been a survival benefit.
Yeah. So the environment has changed a lot. And if you look at the association between the FTO locus and obesity, it doesn't become prominent until after World War II. Before World War II, where we basically were much more active and we didn't have, fast foods like McDonald's, there was almost no association between FTO. So it used to be a fine allele to have. Not only that, but as you mentioned, it may have actually been a beneficial allele as you crossed the desert, as you went through ice ages, etc., to be able to store every calorie in your body. And it could be drift out of Africa, but it could have also been selection for a different time when we actually had fewer calories than now. So I agree with you that it may have actually been a selective thing. So Simon?
Yes, thank you. First of all, fantastic talk. I was just wondering, the other popular topic of the summer school was the idea of generating synthetic data. And the way from those talks, I understood it as learning a representation of the whole mapping, of the actual mapping of the, for example, knowledge behind it. And I can see that you were talking about something very similar, but just with actual distributions. I was wondering if there's any collaboration opportunities between these two fields where you're trying to find, while you're trying to improve the learning result by generating synthetic data from the already learned representation. Yeah, just a curiosity.
When people are training self-driving cars, they just can't have enough data for training. And there's something known as data augmentation, where you can basically take an image, you can crop it, you can rotate, you can flip it, you can change the colors, and it's still a cat for every one of those. So it's still the same label. So you can think of it the same way, where basically in self-driving cars, they're now running simulations of the world to basically feed enough data to those networks. And in biology, you can do the same thing, where you can basically create a model and start running simulations from that model. There's an advantage in that these extraordinary large representation models can now capture subtleties that might not be in the original data. But there's also the danger that whatever you're simulating is what you're going to learn about from the real world, and it might not actually be the real world. But the advantage there is that you're kind of filling in, you're interpolating these intermediate steps that you may not have in your training data. So I think that there's great power to leverage these types of simulation-based approaches by sampling not from the X vector, but from the Z latent space. So you basically sample from the latent space, and that's a lot of what you do with variational tone encoders, with graph-auto, with generative adversarial networks, and with diffusion. You're basically learning how to approximate this latent distribution rather than the original X data. Tim, I don't know if we should break here or...
Unfortunately, we really do have to wrap up. This has just been an excellent keynote, and I think you've inspired many people today. So thank you so much for being here. If you want to answer one or two questions in the chat, do feel free to stay on. And
I will stick around and I will answer on the chat, but I will stay quiet and you guys can move on. Thank you so much, Tim.
That has been so brilliant. Thank you.
Thanks, everyone.
Yeah, and I will hand over to Tim Osterling.
And thank you, Dr.